Okay, welcome. We're going to go ahead and get started. We're here today with Hillard Kaplan, who's from the University of New Mexico, and he's going to be speaking as part of our Behavioral and Social Science Research Lecture Series. And Dr. Hillard has been doing research on small-scale societies over the last 30 years in the South America and Africa. His research has been funded by NSF, NIA, and NICHD. And his early focus was on demographic transitions, nutrition, and diet. And over the last 10 years or so, he's been focusing more on aging in terms of behavioral and physical aging. And so he's going to talk to us today about infection and inflammation, cardiovascular disease, and immunosenescence. Did I say that right? I'll let him introduce himself, but please join me in welcoming him.
Thank you so much. It's a pleasure to be here. Can you hear me okay? Good. Well, I'd like to acknowledge also my co-PI, Michael Gervin, and offer a special thanks to the NIA Behavioral Social Research Division and to the OBSSR, without whose funding none of this work would have been possible. So I have several goals for the talk. The first goal is to say what can be learned from the study of energy-limited traditional societies about aging and health, particularly about what it can tell us about the basic biology of people and what it can offer in the way of insights into health problems in the United States. And the other thing that I would like to address is some of the potentials and challenges associated with multi-level community-based research in collecting epidemiological, demographic, and behavioral data at the population level, and how that can be combined with more in-depth clinical and bench sciences, and the challenges and solutions for conducting research in rural developing world settings. So I'll first be addressing, did people actually live long enough in the past to experience significant aging? And when I talk about the past, I'm going back tens of thousands of years. And then I'll be discussing aging, productivity, and intergenerational transfers in small-scale societies with a little examination of phenotypic aging. And then I'll be focusing the latter half of the talk mostly on new unpublished data on the epidemiology of infectious disease and immunosenescence among the Tsimane, and hypertension, cardiovascular disease, and diabetes. And I'll be concluding with some take-home messages. So this man here is an Aceh. He's a hunter-gatherer. I took this picture in 1981. And how old would you guess he is? Any guesses? He's 70 when this picture was taken. In this picture, he's digging for armadillos with a machete, and he has to go down about 2 meters. So, how long do foragers actually live? So we can think about, given that you've reached a given age, how many years can you expect to live? Well, at birth, hunter-gatherers, these are all the hunter-gatherer groups that we have some data on, as well as you'll see Sweden from the 18th century, and wild chimpanzees. At birth, it's about a 35-year lifespan for humans and 15 for chimpanzees. But by age 15, people actually have an extra 40 or so years of life remaining, which would get them up on average to age 55. And if they make it to age 45, they have an expected extra 2 decades of life. And these are people without access to any Western medicine. So many people actually live into old age in these small-scale groups. And in fact, what's interesting about the life expectancy curve is that all these groups really overlay very similarly on top of one another. as if there is a stamp to the traditional mortality profile. If you look at the death distributions, this is the actual number of people who die, the proportion of people who die at given ages. These groups tend to have their peak mortality at age 70. After age 70, there's so few people who are left, and mortality is increasing to a high point there. And you can see in the United States, it's in the 80s. So one of the interesting things is that most of the differences, or the differences between the mortality rates in these traditional societies and the United States, is really early in life. So you have a 32-year life expectancy at birth compared to a 78. And in the first year of life, the mortality rate for these small-scale peoples is 200 times as high as in the United States. But as we get later in life, it's 14 times 7, 3. And so as old age starts to kick in, there's more convergence, and the basic biology starts to take over as opposed to environment. So we spent the last 9 years working with a group called the Tsimane in Bolivia to investigate the causes of this mortality profile and what is the underlying physiology of aging. So Bolivia is in South America. It's a landlocked country. Here's where our field site is in the lowland part of Bolivia, and the villages that we work in are along the river and in the jungle. These were the first 15 or so villages that we started working in, and then we've expanded to 85. I'll give you a little bit of a feeling for what it looks like there. It's rivers and forests. This is actually one family. You can see that they have very high reproductive rates. Almost all the family's possessions can be seen right here. They hunt and they fish and they grow crops. So plantains, papaya, this is yucca or manioc, rice, and corn are their main staple crops. So why do we pick the Tsimane as a study population? Well, first, the Tsimane live in ways that are really similar to what we can think of as the human past, in terms of having a high pathogen burden with no public health and access to Western medicine, a low energy balance. They have as many kids as they have without attempting to do birth control, minimal health care. They have a traditional subsistence strategy, and but there's still enough of them that we can actually get decent population epidemiology. 10,000 is a small number in some people's view when we think about nations, but in terms of small-scale populations, this is a relatively large size. And the Tsimane also have a gradient of acculturation and market involvement, because up until 1990, virtually none of them were involved in any kind of selling or wage labor. But now some of the villages that are closer to town, they sell stuff, buy stuff, and are much more involved in modern life. So just to give you a little bit of a sense of what we know about the Tsimane, one of the surprising things is that, so these two curves are the amount of food consumed per day by individuals. These are boys, these are girls as they become women. And this is food production. What you can see is the crossover point at which people consume as much as they produce is only at about age 20. So kids are subsidized until that age. And what we see is that production abilities, both the amount of food they produce and the food that they produce per unit time, peaks in the 40s, sort of when we become full professors and then go to pasture. One of the interesting things that we find is that the production stays higher than consumption all the way into old age. And we have been able to actually measure the flows of food between pairs of individuals. So this blue line is the flow of food between dads and their kids. This is what we call the net flow of food, because kids give food to their parents and parents give food to their kids. But this one you subtract one from the other. So at age 40, men are giving about 3,000 calories to the sum of all of their kids. And they give food until they actually reach about 70. Here we see them starting to give to grandchildren. These are women. Women are giving less food because they're spending more time in childcare, and women giving to their grandkids. But we see at around age 70 is where people start hitting that point where they're just about a net zero, neither producing nor consuming more. So we have been thinking about this as a sort of natural age of lifespan in traditional societies, because that's the modal age of mortality, and this is the age at which they stop being productive members of society. And it turns out that usually they're very closely linked at the individual level. When you get really sick and you can't produce very much is when you're very close to death as well. So this is what some of the phenotypic changes with age look like. So these are measuring upper and lower body strength with a dynamometer. And you can see that men are stronger than women, but they're sort of decreasing together. And so by the age of 70, you already got about a 30% decrease from age 30 to 39 in your strength. And in the ability to perform different tasks. So this is a classic task of aging, being able to stand together with one foot in front of the other with your eyes closed, which don't try at home. And what you can see is that women are really decreasing, and particularly we see this fairly big jump between the 60s and 70s in a lot of different measures, low rates of decline until then, and then 70 plus we see bigger rates of decline. This is how much weight in terms of plantains or rice. An arroba is 12 kilograms, or about 26 pounds. So this is how much rice you can carry all the way from your fields. And you see women decreasing with age. These are the trees that people have to chop down to make their fields. And again, we see that men stay pretty good until about 70. And we also see that the ability to carry a weight a long distance drops with age. These differences you can really see depend, in terms of your work, on how hard it is to do. So here are trees of different sizes, really big trees going down to little trees. And the time that it takes to chop one doesn't hardly get changed when they're little trees. But when the work is really hard is where you see the effects of aging. And that's why people can stay productive until reasonably old ages. So the rest of the talk will be more biomedical in orientation. So what we have been doing is we've been going to the villages and conducting physical examinations. This woman is a doctor, and here's a Tsimane translator, and she's being interviewed. This is the patient. And they get basically a head-to-toe physical examination in the field and treated for their illnesses, which are quite varied. Lots of skin infections, This is a girl with tuberculosis with Pott's disease. This woman had really bad skin infection that turned into kidney failure, and she lost her baby at the same time. And this woman came to us with end-stage cancer, and the only thing we could do for her was to give her morphine to help her out. We've conducted electrocardiograms on about 1,200 people, and we brought our laboratory to the villages in the most rudimentary way. Here's liquid nitrogen tank for collecting blood and urine samples for later processing. We also did some in-field processing of the samples. Through time, we've been building our relationships with the communities, and we started off in the first year with 18 communities, and then we started gradually increasing. And in 2010, we made a big switch, and we moved from 25 communities to 82 communities. But what we ended up doing is we brought all the people over the age of 40 to our central clinic instead of treating them in community, so that we could do much more in-depth laboratory research and echocardiography. So it's quite a logistical challenge getting people back and forth or getting to the villages. There's floods all the time, and my headache rate has increased dramatically since we started bringing people in. And this is to give you a sense of what the epidemiology of disease looks like with the Tsimane. About 70 to 75% of people across all ages are infected with one or another intestinal parasite. At any one point in time, a snapshot, 40% of kids have a respiratory infection, but even into adulthood, 1 in 6 people will have a respiratory infection at any point in time, because these are sort of random snapshots. They're not people who are coming to the clinic because they're sick. Gastrointestinal infections and skin infections. Hookworm is by far their worst intestinal parasite that is associated with anemia and drains them of critical energy resources. This is a small view of the serology we've done with antibodies to specific diseases. You can see that yellow fever, hepatitis, most people have been exposed to leptospirosis. Chagas has some frequency, toxoplasmosis, and there have been measles epidemics that have gone through these communities. So this is what the white blood cell counts look like by age. And so the dark line is the Tsimane, and the gray line is levels in the United States from the N. Haynes sample. And the things that I want you to get out of this figure is, first of all, neutrophils, lymphocytes, and eosinophils are much more at much higher levels with the Tsimane, and they decrease with age. And you don't see very much going on in the U.S. sample. The only case in which the Tsimane are lower are monocytes and basophils. But this, if you put all these together, you get an average in childhood of white blood cell counts over 10,000. And that's what doctors usually consider as the marker of an infectious process. Eosinophils are often related to parasitic infection, and that's probably why they're so elevated. So we see that they're high and they're going down with age. Now this is data that shows that there seems to be an interesting age pattern and continuation of inflammation is measured by C-reactive protein. So these are the Tsimane, who are the least acculturated. Here is an Indian group called the Shuar in Ecuador, who have better health care and better public health conditions. They're still quite high in early childhood, but they don't stay nearly as high. And this is the Inchiante study in Italy and the N. Haynes. This is the N. Haynes. So what we can get from this is that Tsimane are chronically exposed to high rates of inflammation throughout their life. And so we want to ask the question, how does exposure to infections, viruses, and parasites shape investments in immune function, growth, and reproduction over the life course? And what are the consequences of these insults and other factors on older adults in terms of frailty, disability, morbidity, and mortality? So here's a figure that looks at the distribution of IgE, which again is related in parasitic infections. What you can see that the Tsimane have a 40 times higher rate of IgE, and the Shuar in the middle. And if you look at the distributions with age, well, these peaks are a little subtle. What's happening is that you get an earlier peak, an earlier acceleration, the higher the exposure is. So the peak age of IgE is around age 8 or so for the Tsimane males, and doesn't really peak with the N. Haynes until later. But you can just see that there is a much higher and intense growth in the levels. Oh, I'm not expecting you to study this figure in any detail. What we see is that when we measure serum cytokines, we see that there is a different pattern of decrease in cytokines before age 40 and after age 40. In particular, IL-10, IL-6, and interferon gamma are increasing more, and then we start to see TNF-alpha and IL-5. The interesting thing that we're seeing here is that IL-4 actually increases with age, and the ratio of interleukin-4 to interferon gamma is thought to represent how much of a bias in the immune system there is towards Th1 responses, Th2 responses. And so what's happening with age is that the response is becoming more Th2 biased, as if the ability to respond with T lymphocytes is going down more. Now, one of the things that you have that's a complication when you look at serum cytokines is, are people with higher levels better able to defend against disease, or are they worse able? Is it because they're sick that they have high cytokines, or do their cytokines actually prevent them? So one of the ways that you can deal with this problem is you can do challenges where you take their blood and then you give an antigen to it, and then you incubate the blood and you see what the cells in the blood that are still alive are producing in the way of cytokines. And we stimulated them. These figures show 3 different types of stimulation. This is LPS, which is a antigen from the cell surfaces of gram-negative bacteria, H1N1, and this is H1N1, and this is Ascaris, which is a intestinal parasite. And you can see again that IL-4 everything is going down with age, again suggesting that the ability to respond to antigens is decreasing with age, and perhaps they're burning out their immune system more quickly than would be the case if they didn't have to mount immune responses and proliferate cell lines as much. But the decline in age depends on which antigen and which cytokine. So IL-4 in response to bacteria is not dropping, whereas interferon gamma, again, is dropping, the same as the pattern of the initial cytokines. Now, if you have a parasite, it actually reduces your ability to respond to infections, and the cytokines that are most involved in viral, most involved in bacterial, are more affected by helminths in response to the bacterial or the viral antigen. So what I'm trying to give you is a sense as a population that is stressed by multiple causes of infection, and through that process, their ability to fight new infections is actually inhibited because they're dealing already with parasites. Now, we've just started, this is from our newest data coming in just the last few weeks, doing analysis with flow cytometry of lymphocyte distributions with age. So we all know that CD8 T cells are what really start to burn out with AIDS. And in some ways, AIDS has some parallels between the life of the Tsimane, in which they're exposed to all sorts of different infectious processes and have to deal with it, and people who have AIDS who are really burning out their T cell lines. And what these are showing is that both CD4 and CD8 cells are decreasing with age. The only one that's increasing with age are natural killer cells. And one of the things that we're seeing as a pretty radical effect is the percentage that are naive T cells, that is, those that can respond to novel antigens and then form clonal lines to novel ones, as opposed to experienced or memory T cells, which are already specified towards specific antigens. And so this is a good indicator of what might happen if you get exposed to new diseases that you haven't had before as an adult versus as a child. And I bring these two figures together for you because this is a figure taken from before our study of the mortality hazards based upon collecting demographic data and then asking people a whole series of symptoms about people when they died and what they died of. This biggest color over here, the blue, is infectious disease, and what you can see is that the probability of dying from infectious diseases decreases till a low point in childhood and then starts to increase. And so we have white blood cells that are going down with age, but the likelihood of dying from disease is starting to go up, suggesting that it really has something to do with an immunosenescence. And you can see that in the US, you don't see very much pattern there. The last topic I'm going to address today is the quote, man-made diseases, cardiovascular disease, and its major cause of death, and disability, and in developing countries, the burden of cardiovascular disease is increasing, and in fact, it's causing 2 times more deaths than HIV, TB, malaria combined. So I just read a paper saying that this is where we should have been focusing rather than here in the developing world if we wanted to lower mortality. And the risk factors for CVD are increasing worldwide. And so by 16 to 19-year-olds, 15% of North Americans have some signs of early coronary arteriosclerosis. And the majority of Westerners over 60 have arteriosclerosis, and some people consider it to be a normal part of aging, so that's why the quote's quoted from a paper. In fact, there's now some evidence of arteriosclerosis in Egyptian mummies, in the pelvic and thigh arteries, and atheromas in the subclavian artery, and it's also been found in Chinese and Inuit mummies. So perhaps cardiovascular disease has a very long history. Well, one of the interesting links that has been increasingly emphasized in recent years is the relationship between inflammation and CVD. So inflammation seems to be implicated in all stages of arteriosclerosis, and inflammatory markers like CRP, which I just showed a few slides back, is highly predictive of both cardiovascular disease and stroke events. And the Tsimane are much higher than Americans in CRP throughout their lives. And of course, an infectious environment leads to high levels of CRP. On the other hand, the Tsimane, because of their active lifestyle, have some protective factors. So read here where it says Bolivia, read Tsimane, please. And you could see these are the proportions of individuals in America that are obese with a BMI greater than 30, or high cholesterol greater than 240. And you can see that the Tsimane are very, very low in both of those measures. This gives you a kind of a better sense of what the distribution of total cholesterol looks like, and low density lipoproteins. And what you see here is a mode in the Tsimane of about 65, 70, which is considered very, very low LDL. And the distributions are overlapping, but clearly separate. And the Tsimane distribution is much more peaked. One of the things that we're seeing indications of is these disease predictors are actually associated, IgE and CRP, IL-6, with some decreases. Some are significant and some aren't. And cholesterol is positively associated with hemoglobin, or if you're anemic, you're likely to have very low cholesterol. So in some sense, we're starting to see that cholesterol is really a good thing at low levels. If you don't have enough of it, you aren't energetically ready to mount defenses. Now, looking at diabetes risk, so these are the glycated hemoglobin levels, where greater than 6 is considered to be sort of on the risk of diabetes, and as you go up, you have diabetes. These two areas are the least acculturated, and here, as we move to the right, are the more acculturated villages, and we're already starting to see some effects on glycated hemoglobin. Now, if you look at the percent that's greater than 6, in the upriver and the forest communities, it's a 0. And we're at 15%. Most of them are in the low 6s in the more acculturated communities, but we're already starting to see effects. Now, when we looked at hypertension, so this is a figure with the percent of people with systolic blood pressure greater than 140 by age for different countries around the world, and the Tsimane are much below the rest of the groups, but we start to see the beginnings of the rise in the 60s and 70s. But most of that rise is actually due to women, and it starts right around menopause. So these are data, it's a funny kind of a graph, but these are taking longitudinal data on blood pressure, so measuring the person's blood pressure year 1, year 2, year 3, and year 4. As you all know, blood pressure is sort of noisy. If you're stressed, your own blood pressure will go up. But so this is the distributions of individuals of these different ages of their rates of change in systolic blood pressure per year. And what you see with the men is that there's no average or median rate of change that's greater than 0. So we're seeing in most men virtually no increase in blood pressure. And the women's ones, their mean is about, a 3/4 of a point per year in middle age. And this is a figure that looks at the intersalt study of different countries that have been measured, and it looks at the slope of increase. So a 4 would be 4 points per decade of average increase in systolic blood pressure. So these are countries, and you can look at their median systolic blood pressure and see how that is associated with a longitudinal increase with age. And these 3 groups are all the traditional societies that show very little increase in blood pressure with age. The Tsimane are the black dot. One of the things that we did was look at peripheral arterial disease by comparing the blood pressure at the in the arm with the blood pressure at the ankle. And if you have our peripheral vascular disease, your blood pressure will tend to be lower at your ankle than it is up at your arm because it's further away from your heart. And 0.9 is considered to be a cutoff for peripheral arterial disease, and we did this with 350 adults. Now this is what the average ankle brachial index is. It's about 1.1. Actually, I said the opposite of what I should have said, is that 0.9, as it goes down that ratio, it's considered worse. What you can see with the Tsimane is there's almost no change with age, whereas in the US it tends to go down with age. And our worst outliers are still above the line. So we found nobody using the ABI index as at risk of having peripheral arterial disease. Part of the story here, we have exercise, we have diet, but we also have this parasitic infection. There was just a new paper that came out in Science with commentaries that suggested that eosinophils may be, and those agents that increase eosinophils, which tend to be in the parasitic line, may be protective against heart disease in the formation of atheromas. We did a quick analysis of eosinophil counts because you noticed earlier how much higher they were. And what we see is that as eosinophil, you can pick your causal arrow here. As people get fatter, they have lower eosinophil counts, or as people's eosinophil counts go up, they become thinner, which may be more the causal arrow. But what you can see is here's again where we start to see these acculturation differences occurring. So the people who are further away have the much higher eosinophil counts. But in both groups, as you get fatter, they go down. In order to pursue the line of reasoning that says inflammation leads to vascular disease, low cholesterol consumption, exercise reduces vascular disease, how much vascular disease do they really have? We started a year ago to measure the thickness of the carotid artery using ultrasound. This is one of our physicians who came to the US to be trained to do this. Now, here's a scatter plot of the, it's actually a lot cheaper to collect the data than to get them analyzed. So we have collected 465 of them, but we've only gotten 76 analyzed. The lines are pretty linear in these scatter plots. We specifically chose a cluster of people at age 40, and then we chose a cluster of people who were older, but since they were new additions to our sample, they all seemed to have the same age until we really investigated their ages, and then they started to spread quite a bit. But you see that, just like in the US, the thickness of the intima media increases with age, slightly more so for women than for men. This is what the Tsimane CIMT looks like in relation to US ethnic groups with data that I took from tables from the multi-ethnic study of arteriosclerosis. And so these bars here are whites, blacks, Chinese, Hispanics, and the Tsimane. And the Tsimane are kind of looking a little bit higher, especially in older ages. So again, this is going in a slightly different direction. Perhaps this inflammation is increasing wall thickness. But when I looked at the percentage of people at different ages with different levels, so an average of 0.9 is considered to be the risk of the development of CVD. Women at age 55 have about a 13% risk. Men only have a 2%. But if you take 1 as a cutoff, which is the beginnings of potential plaques, very few people have them. And if you use the standard definition of a plaque, which is 1.5, there was only 1 person who had a plaque. So that's going to be a story that will be updated with time. But I think that more of the data are pointing in this direction. So if you look at using a step test to look at cardiovascular function, this is 2 ways of graphing the data. One is your heart rate per watt of work, and the other is your heart rate per watt per kilogram to get to control for body size and how much work you can do. And we'll just focus on this one. What we can see is that the US people are in a lot worse shape than the Tsimane are. And you see that both the data only go up to age 50 in the NHANES for this. But you can see this really big slope of increase in the amount of heartbeats you have to do a given amount of work. You don't see very much going on in either the Tsimane males or the Tsimane females, suggesting that they're in better cardiovascular shape on average than we are. And when we did our EKGs, our echocardiograms, we saw a whole bunch of abnormals. And the abnormals were bradycardia, the majority of them. Bradycardia is low heart rate. So you're resting there and you have the things on and you have a heart rate of 60 or below. Well, that can either mean you got something wrong with the electrical conduction in your heart, or it can mean you're in really good shape. Which is it? Well, the data seem to suggest the good shape hypothesis is correct, because men work much harder than women and they are more likely to have bradycardia. But most importantly, bradycardia decreases with age. So as you get older and in worse shape, heart rate, there are fewer and fewer people that have low heart rates. This is what some of our results look like from doing the echocardiograms. So hypertrophies are increases in the thickness of the walls due to having the heart to get bigger to do more work. But hypertrophies with ECGs also are a little bit hard to interpret sometimes without echocardiograms to know, again, whether these are the athlete's heart or a heart that's in trouble. What we did see was a fairly high frequency of conduction blocks, which could be a sequela to Chagas disease because it's associated with it. And so here we may be seeing that some of the heart problems that exist are going to be due to infectious processes. Now, the infarcts is interesting because two different cardiologists got two very different rates of infarcts for us, but there was always a much higher ratio of women to men, which either means that women survive their infarcts more and all the men that had them were dead and didn't make it into our sample, or that they're more common with women. But I think that this number is going to be really reduced when we get our echocardiograms read to really look at wall motion, because the ECG is just your first line of defense in determining it. So I'm going to wrap up now, and I know I did a whirlwind of data presentation with sort of not taking the time to really interpret each slide in depth, but I wanted you to get the big picture here. So we can say that the Tsimane have very high levels of inflammation throughout life, particularly very high cytokine levels at young ages. But they show little evidence of hypertension, diabetes, and vascular disease. So that the relationship between inflammation and vascular disease may be variable and may depend on other things like exercise, adiposity, cholesterol, calorie balance, things yet to be determined, such as parasitic infection. That there seems to be some indications in other science suggesting that the Th2-based immune system may also be cardioprotective, and the Tsimane relative to Americans show a higher Th2 bias, and that in general cardiovascular disease may not be an inevitable consequence of aging. I think heart aging is, but the vascular component may be a very minor part of the causes of death in human history. But that chronic infection does appear to have long-term consequences, both in terms of immune function and organ damage due to disease, and that immune system aging may be important player in the old age death in high pathogen environments. So if you compare back to the early figures where I showed what do the mortality rates look like in pre-Western medicine societies, they seem pretty similar. And there may be a basic biology of aging that's giving that timing. But the pattern of disease and disability seems to really depend on the environment, what people are exposed to, and what their lifestyle habits are like. So, given the focus of this lecture series, I'd like to just conclude with some comments about what are the opportunities and challenges for doing research in the developing world. I think that it really expands the range of variation relative to the US, and by doing so, as you move ranges, you sometimes get new insights into what seem to be a very tight causal link between things that then doesn't have the same relationship. So there can be interactions as you move across range of variation. And looking at people who aren't overfed, who are still working a lot and still are engaging in uncontrolled fertility can probably give us some really fundamental information about human biology, because after all, 99% of human evolutionary history occurred in small-scale, pre-farming, hunting and gathering groups where marriage, brain size, the aging process all seem to have been largely shaped prior to the emergence of agriculture. And because communities need health care and are interested in participating in studies that provide health care, it allows you to collect information on behavior and disease status at multiple levels. You can go into their communities, you can bring people to clinics, and it enables you to combine larger scale population level demography and epidemiology with more in-depth laboratory and clinical investigations. And this is a program that I think could be exported, where you get a lot of large-scale population data, and then you begin to do more focused, smaller sample, but in-depth studies. And so there doesn't need to be this gulf. Of course, it does raise multiple challenges for you, because the political landscape is always changing. People don't like to be just subjects, so you have to deal with the social aspects of becoming a like member of their community. And of course, they present horrible health problems to you that if you're going to study them, you have to figure out a way to help them with. So some of the solutions to the challenges are, I think you really have to satisfy healthcare needs. This should be part of a normal budget for doing research in conditions where people can really use healthcare. And in the developing world, healthcare can actually be quite cheap to deliver. You, of course, have to build trust and social relationships with people. That's why we couldn't start our study with 85 villages. We had to start small. And you need to form all sorts of networks in order to be able to pull off not getting kicked out of either the communities or the host country. And satisfying the healthcare needs is usually much more doable cheaply if you have good relationships. We have relationships with nuns, with Christian organizations, anywhere we can get help, because once we start getting cancers or really big surgeries, we need to look outside and not use NIH funding, but you can pull this off. And doing human capital investments in local people, getting them on board, teaching them health promotion, and giving them research training. I have now trained maybe 20 different Tsimane in collecting anthropological and health data, and they see it as a road to upward mobility for them. And you don't necessarily only have to do things that are related to your research in order to develop the relationships that will then allow you to collect large amounts of data. And I'd just like to conclude with one last point, which is that what I've seen with lots of larger scale demographic studies, there is, okay, well, we have to collect a biomarker here or there to get our biology information in there and link to our social information. But I've met with lots of researchers who believe that things can't be done. It's too hard at the population level to conduct bench lab work. And what I found is that you think a lot about it. There are logistics that can be pulled off to combine working in the field with getting deeper answers. And with that, I'd like to thank you all very much for listening to me for an hour. Happy to take any questions. Yes.
Can you talk a little bit more about reproduction and fertility in this group? Because, you know, in that picture you had, they had a whole lot of kids, and yet they've got some serious health problems, it sounds like, which you might expect would impair their fertility. And also what their patterns of, say, lactation and nursing are like women, starting out.
Yeah, I could talk about that for a while. I worked on those issues prior to working on these issues and couldn't fit them into the talk. So the mean total fertility rate for the Tsimane is now about 8.5. And I think that you're right, that at the individual level, things like anemia, BMI, make a difference to parity progressions and to infant survival. We have good data showing that it makes a difference on the likelihood of moving from one kid to the next, how long it takes. But, you know, one of the interesting things about Indian peoples in South America, as fertility rates have gone down through the rest of the country, it hasn't happened. Demographic transition and most of the small-scale Indian populations. But what has happened is some health care, some change in diet. And so the two together, you're getting a lowering of the mortality rates and maybe even a slight increase in the fertility rates, leading to population growth rates that are absolutely astronomical, like 3.5% per year with 20-year doubling times. And that's what the Tsimane are experiencing. And there seems to be evidence of maternal depletion with the successive. I didn't show you that I took out at the very end, before trying to do my slide reduction act, the data on teeth loss. But people are losing teeth through time, so that by the time you're 60, you have an expectation of about 10 teeth in your mouth from 32. So, and women have a much higher rate of tooth loss than men. That is likely associated with the burden of gestation and lactation. So what I guess my thought is that it's part of what's keeping the BMI lower, is that there's just a lot of mouths to feed and a lot of reproduction going on. And what women are experiencing now is a desire for birth control, but not birth control to bring you down from 7 or 8 kids to 2 kids, but just to kind of stretch it out a little bit more, so you don't have 1.5 year birth spacing, so you could have 5, 6 kids under the age of 10. That is really, really a stressor on women. Yeah?
I assume that this is a very genetic and homogeneous group. What could be the
The answer to that is I have absolutely no idea, but it's certainly on my big question list. We've collected a lot of genetic data. We haven't had any money to analyze it, and now it's not the best time to be doing it. There is admixture, but not that much. And what we would like to be able to do, what I think is one of the great areas that this group could add insight into, is the relationship between genotype and phenotype. So we have these studies that are going on that are looking for genetic markers that predispose you towards heart disease, genetic markers associated with C-reactive protein production, for example. So I think there are 2 questions that one could ask. The second one is a little more interesting to me than the first. The first question is, how much of this pattern are we seeing is due to special differences in their genetic distributions? And the second question is, how does the relationship between genotype and phenotype change when you have high disease loads and low energy balance? So some of the genes that today may be associated with cardiovascular disease, under those, the same gene under different circumstances could actually maybe help you get through bad nutrition. And so that's where I think we may end up understanding why some of these genes that are maybe bad in this context exist in the first place, because under other conditions they may have their benefits. Yeah?
In collecting the samples for storage and eventual use in genetic or genomic studies, what do you actually say to the people? How do you explain that process in your informed consent process?
We talked about this at lunch, actually. So we have a multi-level process where we first hold discussions with the Tsimane Council, which is the leadership organization for the tribe, and explain to them what we're wanting to do. And from that initial set of discussions, we learn a lot about how to talk. And then we go to each village and we have a community level meeting in which we discuss all the procedures that we're going to do and why. And if it's something like genes, we do use the kinds of things that would be similar to what might be used here, where you start with the idea of parent-offspring resemblance and some characteristics that they can get their minds around. But what we've found is that these community level meetings is where some of the best communication goes on, because people are having little side conversations explaining to one another. And then we do a third meeting at the individual level with each person and explain to them what we're going to do. But my own experience is it's the second level meetings in which the informed part of informed consent is really occurring. At the individual meetings, there's a little bit of a sense of being overwhelmed with the information. And so we really like to make sure that the community meetings are held before the individual meetings, so that people have had a chance to ask their sister or their brother, what were they saying? And get it through that. You asked, and then you've had your hand up like 4 times, so I finally wanted to recognize you.
Thank you, and thank you for the talk. I just wanted to note that, of course, there is an increasingly common appreciation for like methodology and disadvantage and hard to reach populations, etc. So I wondered if you could just briefly share some of your successful methodologies incorporated into your work with this particular.
Yes. So I talked about all of the things that we do for the ethnic group as part of the trust building. But where I've found the creativity and the flexibility to come in a lot is in logistics. So this is why we switched from bringing our people to the villages to bringing people from the villages to our clinic, and having a mixture of those two. Because bringing people to a clinic requires a whole logistic. I have trucks, I have boats, I take people 3 days, first by river and then by truck to our clinic, and then I have to feed them and I have to bring them all back. But once they're in our clinic, they're like, okay, pass me through the ringer, and they go from station to station. They like our food, they like our house, and then they like the fact that they're going to go home with some gifts. And they've also had a chance to see town for free with their transport taken care of. But actually, all the aspects of those logistics, from when we were going out to the field, we had to be incredibly creative. You know, most people were doing blood spots. We realized that we couldn't do as much with blood spots as we could with serum. And so we got liquid nitrogen, centrifuges, solar panels, and you know, you have 3 months of storage with liquid nitrogen before then you can get it to freezers. So as we were developing our study, each step required that we think, can we really pull this off? What will it take socially to pull it off? And what will it take logistically to pull it off? And then politically as well.
So assessing the on-the-ground reality and thinking of how you can adjust and address them based on...
Exactly. And starting not so with not such high goals, and then as you get better and better, you crank up your sophistication of what you can do.
Do you see dementia in this population, and do you have cognitive aging as a phenotype?
Okay, so there's the first answer is yes. And our Tsimane translators tell us, oh my God, this interview, this person was, drifting, lost, and and we have measures of cognitive aging that have to do with being able to follow a sequence, so taps, where it was, things that don't require writing ability. But I'm unhappy with as far as we've gotten, and this is where I think the cultural gap is requiring a level of creativity that I haven't gotten to yet, because administering, for example, the more that you get conceptual, not stuff like how many animals can you remember the name of, but deeper logical reasoning and higher level cognitive functions. That's where I feel that the linguistic barrier we haven't been able to surmount. And rather than do stuff that I think has very questionable validity, I've been holding off on that, because
you don't think the draw clock is going to work with this?
that you can do, but it's just, is it really going to get us the information that we want? And we did this with measuring of depression, and I just felt like we were getting garbage in, because what one person would say versus what another person would say. When you try to use a Likert scale with people like the Tsimane, and it's even really hard to translate a Likert scale into more than 3 or 4 levels. One gets a sense that they're just sort of, based upon how they understood, they're going to put themselves in some part of the continuum, and the mapping between what you wanted to know. And in general, as an anthropologist, my experience has been, start with stuff that you don't need words for, that you can measure, and then as you get better and better at the language, then start to move into those things. But I have yet to find the measures of dementia that I think we can apply yet. But any help I could get, it would be.
Last question. I'm struck by your list of solutions that, in some respects, have provision of their own services are no longer serving the needs, and those interventions might impact a very measurable percentage, whether it's cytokine levels or parasite levels. So do you have any observations of cohort effects that you see in infections or the parasite levels?
This is a promissory note that I have in terms of the analysis, because as you can see, we started in some villages later than other villages. So we can compare where things are in the later villages to where they are in our own village. And there's clearly an uncertainty principle going on here, that the act of studying people requires that you change them. It requires it for both logistical and moral reasons. You can't have somebody come to you with a serious health problem and have the ability to fix it and not do anything about it. And people wouldn't come to us if they didn't see that we were a source of help. And so it absolutely behooves us to measure our own effects and to find creative statistical means, because why are we there? Well, we're there as part of a whole process of the new world entering the lives of people. So we're not the only factor. This train has left the station and is moving with all sorts of things, including us. Their diets, their ability to access markets, Now with a new socialist government, there's more interest in getting them health care. And so when we have all of our different villages put together, it's really going to enable us to start to say, okay, when we were there versus this many years later, and then look from this one this many years later, and then be able to try to figure out. I think there's absolutely no doubt that we've saved the lives of a lot of people who would have been dead if our study wasn't there. On the other hand, because we're working in 85 villages, we only get to each village once a year, so they have the whole rest of the year to die, unfortunately, and have all these illnesses. So, but the next time they've come around, so for example, you give anti-parasite medication, a year later there's no difference from the year before in how many parasites. That may not be true for some of the other things that we're doing, and so it's a really important question you're raising. Well, thank you very much.